as was mentioned before, we are interested in how prenatal infection represents a risk factor for neurodevelopmental disorders. So it is known that there's like an early life programming of health and disease, and that also environmental influences that happen during early development can result in permanent changes, and that can, under certain circumstances, increase the risk for developing certain diseases in adulthood. And one such environmental adverse influence is maternal infection. Maternal infection was recently again moved into the center of public attention by the recent outbreak of Zika virus in South America. And we actually have clinical, preclinical, and epidemiological evidence that maternal infection represents a risk factor for psychiatric disorders. So if you're exposed to an infection during prenatal life, this can increase your risk of developing psychiatric and cognitive disorders later in life. And this phenomenon does not seem to be pathogen specific. It has actually been shown for a variety of viral, bacterial, and also protozoan pathogens, and it can increase your risk of developing depressive disorder, bipolar disorder, schizophrenia, or also autism. And those diseases actually do share overlapping symptoms and also common underlying risk factors. So despite the fact that the link between prenatal infection and developing certain psychiatric disorders later in life is well established, what we don't know is what are the mediating mechanisms. And there's a lot of pathophysiological processes correlated with a prenatal infection, which includes also macro and micronutrient deficiency, but of course also soluble immune factors. There's also an increase in reactive oxygen species and other endocrine factors that can partly be transmitted via the placenta to the fetus and lead to fetal inflammation and oxidative stress. They can also, even if they're not directly transmitted via the placenta, actually interfere with correct placental functioning, lead to placental inflammation, insufficiency, or also altered placental microbiome. And all those factors can work together to interfere with normal fetal brain development and physiological development. And what was or is very much in the center of an important mediating mechanisms is actually soluble immune factors such as cytokines, which then can cause fetal inflammation in the exposed offspring. So as you can see, we have a variety of possible mediating or underlying mechanisms, and so people are interested in developing preclinical models to actually further be able to study the underlying mechanisms. So models were developed to mimic or to study what is happening between this maternal immune activation in your prenatal life and then leading to brain and behavioral development changes in the offspring. And there are different models, and the one that we work with and that is also represent the data that I'm showing you today is based on that model, is the so-called poly-IC model, which I will explain in detail later on. But there are also other models, for example, working with LPS, mimicking a bacterial-like immune phase response. Turpentine model is also quite well used and established, where you create the local inflammation with administering turpentine intramuscular. And recently, people are also working with selected cytokines, such as, for example, the model of IL-6 administration. So as I said before, in our lab, we're working with the model of poly-IC. And we are working with mice, but I want to mention that this model has also been established in rats and also in non-human primates. And so what we do is we administer poly-IC or vehicle control solution to pregnant dams. And poly-IC is synthetic double-stranded RNA, which is recognized by toll-like receptor 3 and leading to a viral-like acute phase response in the pregnant host. And so there are different varieties, let's say, of this model. You can either mimic a very intense immune activation with a high dose of poly-IC, where you also, which leads to a high inflammatory response in the maternal host and also in the fetus. And this factor alone is sufficient to induce long-term brain and behavioral changes in the offspring. We have also established models where we use a low dose of poly-IC, which per se does not lead to behavioral changes in the offspring, but then if you can compare it with genetic or environmental risk factors of interest to see if it interacts in producing long-term brain and behavioral abnormalities in the offspring. Today I will focus mainly on this model where really the immune activation per se already produces behavioral changes in the offspring. And while the poly-IC model was started off in 2 labs, it's now widely used and established, I think, in more than 30 labs. And this is also due to the fact that the poly-IC model is capable of inducing a wide variety of behavioral deficits that are relevant to psychiatric conditions. So what you can see here is poly-IC exposed offspring develop deficits in sensory motor gating, which is usually measured in preclinical models, but also in psychiatric patients with pre-pulse inhibition test. The mice usually also display attentional deficits, a deficit in social behavior, in exploratory behavior, and impaired working memory. So you can see a wide variety of behaviors that you can assess in preclinical models and that are relevant in psychiatric conditions are caused by poly-IC models. And this is, I think, why it's also very widely used nowadays as a preclinical model. So as I said before, it's not really known what are the mediating mechanisms that lead basically from a very early life insult, such as this infection or inflammation in prenatal life, to long-term neural abnormalities that you see in adulthood. And in recent years, there was a lot of interest centered on epigenetic modifications, since people think that this is one of the mechanisms how environmental adversities can actually lead to long-lasting changes. And so epigenetic just refers to changes in transcriptional activity without changes in the DNA sequence itself. And there's a variety of epigenetic modifications, such as, for example, DNA methylation, also histone modifications, or RNA expression patterns, such as microRNAs, for example. And we also wanted to look into potential epigenetic changes in our model, and we chose an untargeted approach and assessed genome-wide DNA methylation. And what you can see in this heat map is that a lot of genes are actually hypo, but also hypermethylated in poly-IC exposed offspring as compared to control animals. When then you analyze those differentially methylated genes in pathway analysis, it revealed that indeed a lot of those genes are important in processes such as synaptic development, myelination, or development of neurons. And this is just an example from our lab, but also other groups have looked into epigenetic modifications following prenatal immune activation and also found changes in histone modifications or microRNA expression patterns in brain regions relevant to psychiatric diseases. And I forgot to mention that what we look at here is DNA methylation in the prefrontal cortex of those animals. And epigenetic modifications have also been a lot of in the center of interest when people started studying how environmental adversities can actually not only affect the directly exposed subjects, but can be transmitted over generation. So this is an upcoming topic. So how important is actually transgenerational epigenetic inheritance And given that now we know that we do have epigenetic changes in the brain of poly-IC-exposed offspring, we were wondering, do we also have maybe an epigenetic transmission? So do we have transgenerational consequences of this prenatal infection? And to address this question in our model, we decided to produce F1 offspring. So as I said before, we administer poly-IC control solution to pregnant mice. And we do a behavioral phenotyping once they reach adulthood. And we took a subgroup of mice that we intercrossed to produce second generation offspring with either a poly-IC or a control history. And we subjected those animals to the same behavioral phenotyping. And I'm just showing you an example of behaviors that we test in those mice. So here you can see social interaction test, where basically a mouse, it was the 3-chamber test that was shown before. A mouse can just choose if it wants to interact with a mouse or with an inanimate dummy object in our case. And you can see that a healthy mouse would always show a clear preference towards interacting with the mouse. And this is reduced to chance level in poly-IC-exposed offspring. And when we test for social behavior in our second generation offspring, we find the same effect is still true for second generation offspring. We also performed tests for sensory motor gating, which sensory motor gating deficits are also present in a lot of psychiatric conditions, and you can basically measure the same reflex in rodents as well as in humans. And you present start eliciting pulses and pair them with different pre-pulses, and usually this would lead to a reduction in the reaction that you showed to the start eliciting pulse. And you can see here that poly-IC-exposed offspring indeed do show a deficit in sensory motor gating, but when you look at second generation offspring, this deficit is absent in second generation, indicating that yes, we do have a transgenerational transmission of the phenotype, but moreover, it seems to be modified when it transferred over generations. So in a next step, we wanted to find out whether it's actually the maternal or the paternal history that drives this transgenerational transmission of a phenotype to the next generation. So we tried to split lines. So we were using either a poly-IC exposed female and crossed it with a control male, or vice versa, a poly-IC exposed male crossed with the control female to produce either maternal line second generation or paternal line second generation offspring, and performed the same behavioral testing. And very much, or a bit to our surprise, I have to say, what we found is that it was mainly via the paternal line that the phenotype was transmitted to second generation offspring. So last, if you want to talk about a phenomenon of transgenerational inheritance, people will always make you study at least 3 generations. And this is because basically you could argue that when you administer poly-IC in pregnant animals, here the fetus is exposed, but it's also its primordial germ cells that it's carrying that are exposed, which give then rise to the second generation. So if you want to rule out that it's an effect of the direct exposure, you would need to produce a third generation offspring, because this is basically the first generation where you did not have any exposure to the initial environmental insult. So we also produced third generation offspring, and we found that the behavioral deficits that we see in the directly exposed offspring are still present in third generation. So basically, if this would be like your father's grandmother that was exposed to an infection, which still would affect certain behaviors. So to conclude, we know that prenatal exposure to inflammation and associated pathophysiological processes can indeed induce long-lasting impairments in various brain and behavioral functions. So we know that also the intensity matters. As I said, there's an intensity that per se is sufficient to induce behavioral deficits. but there's also dosages where you don't see anything, but it can interact with other genetic or environmental factors. And it seems that also the timing when it happens in pregnancy matters in the specific phenotype that you see in the exposed offspring. So as I said, the effects of prenatal inflammation can be magnified, but we should not forget could also be attenuated actually by genetic factors or other environmental factors. So there's a high importance of gene environment or environment interactions. And recent findings highlighted that epigenetic modifications could provide a molecular mechanism for pathology, not only in the direct descendants, but also in their subsequent generation, indicating the environmental history of your ancestors also matter for yourself. And so with that, at the end, I would like to acknowledge our lab members, of course, my supervisor, Professor Urs Meyer, our funding sources, and our collaborators, and thank you for your attention.